Ready to invite the first keynote speaker on the stage. Let me introduce you to Senior Director Jeyaseelan Jeyarj of Asia Pacific Healthcare Sciences and Global Business Unit of Oracle Corporation. He's going to talk about embracing the future key trends impacting the biomedical industry in the post-COVID world.
Good morning. Very delighted to be here today. So what I'm going to do is I will share some of the key trends that's really impacting the biomedical industry as we move beyond the COVID-19 pandemic. So the first thing is, I really see this pandemic as an inflection point in terms of adoption of digitalization. If you really look at the last 20 years, what really happened in the industry versus the last 2.5 years, there has been significant change in the way that biomedical industry has really adopted digital technologies. So that's really fundamentally, I think this will continue to be evolve. As we move forward, the industry will adopt more and more transformation in terms of digital adoption. So this is really evident when the way look at how the treatments and therapies are delivered. And they are becoming more preventive, more curative. It's also becoming more digitized. I really see that at least there are 1 or 2 digital components embedded as part of the digital therapeutics, really focused on medication adherence. For example, like lifestyle changes, how do we really use digital therapeutics to really address some of the complex challenges. Also see this, COVID-19 is not just a challenge for the biopharma industry, but it's also unprecedented opportunity to really transform the way how we are doing business, changing fundamentally the business models, becoming more flexible, agile to really kind of address the key challenges faced in the industry. Now, if you really look at, at the heart of achieving all of this is data. So I really think that data is a critical enabler of this digital transformation. Now, if you really look at now, there is explosion of data. So we deal with large volume of data of different types. For example, like structured data, unstructured data. We also deal with images. We also have sound. Each of these data has its own semantic context as well, right? We also see the data in terms of varying frequencies. We are kind of moving away from data points to a data stream. So those are really the changes that is really evolving. And the data is also becoming periodic, and it's also becoming very sophisticated. For example, a scientific, literature in a natural language form. So those are fundamentally the challenges that we really see from various types of data. So what we really need to do, how do we really bring all these together, unify them, and really make it available for everyone to really collaborate in terms of finding new discoveries? I think that's where the challenge really lies in. So we really see this, data as a key lever that supports much more agile organization to changing market needs. So what I'm going to do, you see here in this diagram, there are a number of use cases. So where we really see the opportunities in terms of going forward beyond the pandemic, right? So what I thought was it's interesting for the participants to understand, some specific examples of some breakthrough innovations that's really happening globally. They're ranging all the way across the drug development life cycles, from scientific research to a preclinical development, to a clinical development, to post-marketing. So really, it's across the spectrum where you really see this thing really happening. And all of these innovations are ultimately resulting into how do we increase the ROI from a research point of view, right? How do we develop more drug candidates? And then how do we really make faster discoveries? How do we reduce the time to market from a drug discovery standpoint? So these are fundamentally the key value drivers that's really, some of these use cases would really kind of play a key role. So let me start with scientific research. So here's an example of what University of South Carolina has done in partnership with Ellison Institute. So what they've done is the recent breakthrough has been to train a predictive algorithm to support cancer diagnostics. more specifically on the breast cancer diagnostics. So many of you are aware, I mean, now we see a lot of adoption of artificial intelligence, natural language processing, machine learning, all of these are really kind of getting adopted in the biopharma industry. Now, the biggest challenge is we need large volume of data to really make any meaningful inference. For example, like if I have to drive a significant relationships, then I need large volume of annotated data sets. So we all know that, in a cancer pathology space, it's really difficult to always get an ideal data set. So what this, USC and Ellis Institute, what they've done is they kind of got around this challenge in a way by using two-step process. First one was to priming the algorithm to find the unique patterns in the cancerous tissue. So that's the first part they did. And then how to train the algorithm to identify the diagnosis. So these are the two-step process they really followed. The first process, what we actually call is tissue fingerprinting. Now, tissue fingerprinting is now really kind of evolving. So what is tissue fingerprinting is actually, it is how do we distinguish the architectural patterns in a tumor tissue that an algorithm can use to distinguish between samples, because no two patients' tumors are same, right? So how do we really use, these technologies? For example, like, they actually use this for 3D organization of tumor to really kind of set up the entire representation of cancer tissue. So what, the outcome of this particular, the innovation is that the AI actually spotted the fine differences, structural differences in the cancer tissue, especially in the pathology slides, with a greater accuracy and reliability than even a human eyes can really spot. So that's really fundamentally the challenge, the innovation that Ray brought in. This also allowed this institute to provide accurate predictions of clinical subtypes of breast cancer. So, as we really see this, becoming more and more adopted, you will find how these kind of technologies really getting adopted, from a scientific research perspective. So let's move from scientific research to a preclinical development. So, we always know that there's always 90% failure rate, especially when pharma companies developing new medications, right? So how do we really address that? So typically what we do is drug compounds are screened in a lab using human cells, right? And then it is tested in animals. This is a typical process that we really follow. But the challenge is that these methods don't always replicate how drugs affect human organs in a real life. So that's been the challenge. How do we really address that? In many cases, drugs will have unexpected toxic effects in tissues which are not targeted from a drug on themselves directly. So what Wake Forest has done is they actually created 3D organoid models. It's basically human organs on a chip. It's a micro-sized chip what they actually created where they're growing real organs on a plastic substrates. So again, if you really look at what they're growing real cells of lungs, liver, kidneys, all of that, using 3D printing techniques. So testing the candidate active ingredients on these human organoid models is actually it's a very effective way to measure the toxicology, getting the early insight of the safety profile than what we typically do for animal testing. So that is the real innovation what they have really found out. So they're able to even identify if a drug doesn't have the appropriate topological profile. As they do these tests, they're generating huge amount of data, for example, like assay data, all of that. So that's really finding it very effective mechanism to understand the safety and efficacy of the particular drug candidates, in the preclinical stage. So now let me just talk about another example. So again, this example is actually very focused on cardiac arrhythmia. So we also know cardiac arrhythmia is actually malfunction of heart's electrical system that makes heart to beat faster or slower or irregular basis, right? But however, we still do not know what is the real cause of arrhythmia. Although there has been a lot of research for the last several decades, still the scientists are trying to solve some of the complex challenges that's faced specifically for this particular thing. So what ELEM is a start of biotech. What they've done is they created 3D models of heart, and then they applied simulation which will aid into the research and the treatment and also find out this exact specific location where the pacemaker electrodes can be placed. So they really pioneered this model approach. How do we really use virtual hearts and then train the system and using simulation techniques to find the real cure. So what it does is it really helps to have at least 50% cost savings in terms of traditionally how we do testing, and almost 15% of improvements when compared to the traditional animal testing. So really, I mean, these are some of the remarkable innovations. They're still in the early stage, but I definitely think that it will really go forward in a significant way. So this is the statement Gartner made. This is pre-pandemic stage, right, where they really talked about how do we really control the cost of the trials. I think this is one of the fundamental challenge all of the industry really has to face with. So how do we really solve this? Right? I mean, how do we really reduce the cost of the trials? One of the way is actually, adopting decentralized trials. I think you have heard this term very often: virtual trials, hybrid trials, decentralized trials. I mean, this is really not new. It's there forever. I mean, it's been there in the industry for more than 5-6 years now. But the pandemic really accelerated the adoption of decentralized trials in a clinical research. And also, the regulators also now have a very good clarity in terms of how they will accept data if they use decentralized trials. So really, it brings in significant benefits. One, it puts patient at the center. I mean, if you look at traditional clinical research model, the patient goes to the site. There will be a principal investigator at the site who will kind of collect all the necessary data. This is how the trials are done, for the last so many decades. So this, what it does is it actually brings the trials back to where the patient is. Patient can be in home or workplace, wherever he is. It gives a lot of option for the patients to participate in a clinical trial based on their convenience. So one is obviously this will really, help to reduce the trial cost. It brings in a lot more convenience, including the drug supplies. For example, the investigational drug product that is used in a trial also has been sent to the patient home, what we call as direct-to-patient home supplies model. So these are some of the things that we really need to look at. And basically, it also need to address the complex operational models. How do we really bring these technologies in a real-life scenario where the data coming in electronically from the patient home, how the investigator will look at the data, all of this has to be really streamlined. I think this is where now the industry is in the early stage of adoption of decentralized trials. So here is a survey that we have recently done in partnership with Informa. So where we talked to almost 257 participants globally. They are ranging from biopharma companies, medical devices, CROs. So we really asked this question in terms of what do you really see based on the adoption, what are the newly adopted approaches, what is the highest responses that you want to kind of share. So clearly, remote monitoring has been evolving as one of the key trends that in terms of adoption. Secondly, we also see a lot of video visits. Say, for example, if an investigator needs to speak with the subject, they can actually use telehealth and video visits. There is concern. I mean, in the traditional model, patient has to travel to the site just to sign a consent. I mean, that's really, how much time has been wasted. So how do we really use electronic consent so that you can really look at all this visit schedule and see which visits can be avoided using electronic technologies. So I think in terms of feedback, we thought like almost 82% provided positive feedback in terms of adoption of these technologies. So the benefits are clearly, one, more timely data, improved flexibility for patients, and also speed of the trial. So you can really do things at scale, at speed. So that's where we really think this is really going to help. So now let's move into the clinical development. So this is the case study about recruitment and engagement of subjects at speed. So the US government actually wanted to recruit 1 million people, what they call as Project Warp Speed. This is at the early stage of the COVID-19 is kind of spreading. So this is a collaborative initiative between NIH, CDC, and the US FDA as well. So they really want to build a platform wherein they want to kind of recruit patients, do a pre-screening platform, where then they can put the patients in a waiting room using digital technologies. So the platform supported the recruitment of 600,000 subjects in a span of 6 weeks. So this is the largest fastest clinical trial recruitment drive in history. So you can really see how they really changed how the traditional model in terms of patient recruitment. We all know that only 5% of the population knows their conditions and what trials are really suitable for them, right? Globally, if you look at those 5%, every 100 people, only 7 people actually make it to the final stage of the randomized clinical trial. So there's significant dropouts in terms of patient recruitment. And the enrollment cost can be up to 40% of the trials, right? So the drug cost is really directly proportional to the trial cost, right? So 40% of the cost is largely on the patient enrollment. So almost one-third of all the delays in phase 3 studies are due to enrollment issues. And also, if you look at the cost per day, is almost $8 million based on the patient enrollment issues. So now we have the technology solutions, what we call as digital patient recruitment, right? So we really think, how do we really bring in a pool of population of data from the social media, from the electronic medical records through a de-identified fashion. We also can look at the disease registries, genomic repositories. How do we bring all of these together and then put those data and the subjects into a waiting room? In the waiting room, you can create the cohorts of patients with the similar traits, similar inclusion-exclusion kind of criteria, and then constantly you update the information on a regular basis as the patients kind of getting enrolled into the platform. So this also gives greater access to the sites to really look at the longitudinal health records of the patients, to really know what medication, what allergies, their demographic criteria, all of these they can really have, look at the information. So these are really some of the things that is really transforming how we enroll subjects into the clinical trials. So now I'm just going to talk a little bit about post-launch, right? So this is an example called vSafe. vSafe is actually an after-vaccine health checker. It was created by my own organization for the US CDC, Center for Disease Control. So vSafe is actually a mobile device where anyone, anywhere can report how they feel after taking a vaccine. On a daily basis, on a weekly or monthly basis. So this has over 150 million anonymous population data that's stored in this platform. So this become a vital source for all the healthcare professionals to really look at what are the common adverse events, how the patient is responding to specific vaccines. This was widely used by US FDA for vaccine approval. So the wide-scale adoption of vSafe actually makes it the largest real-world patient data gathering platform in a single therapeutic area. So this really brought in 4 significant benefits. One is to really evaluate the safety and efficacy of mRNA vaccines, especially for pregnant women. If you really see the initial trials, right, the pregnant women were never part of the trial. So they really don't know what could be the potential adverse events for pregnant women to take COVID-19 vaccine. But there are many pregnant women taken this vaccine accidentally, without knowing the adverse events. But all those data were there in vSafe. So that really helped US FDA to provide the approval, to approve the vaccines for pregnant women. Secondly, the formal approval of Pfizer-BioNTech vaccine, actually, they relied on a lot of vSafe data for that. Similarly, for approval of booster doses by US FDA, right? And fourth, this vSafe data is also been used for several longitudinal studies to really understand a much more precise manner we really see. So now I'm going to talk a little bit about safety. I think this has been been a major focus after this COVID-19 vaccines really been in the market. What is the safety? Do I get adverse events, right? So technology is really now becoming a game changer in the way how we kind of manage safety as we kind of move forward. So if you really look at, on an average, almost about 30 to 50% increase in terms of safety cases that most of the biopharma companies are facing now every year, right? So how do we reduce the speed of processing the case by almost 50%? So this is where the AI really playing a key role. So if you look at safety, there are 2 parts. One is how do we process the safety cases? And then once you have the safety data, how do I find the signal to really understand the risk for the potential population, right? So there are a number of use cases I have listed down in terms of intake. How do you process the safety cases? Then you can also look at how do I do the assessment, for example, causality assessment, seriousness assessment, expected. All these are typical process that's been followed in the biomedical industry. Similarly, if you look at from a safety signal perspective, how do I combine multiple data sources, for example, like scientific literature, health records, and then find the quality signals based on the data that you will really do. So these are some of the example and use cases where I really think the technology is going to play a key role. This is an example of processing safety cases, right? So we know that adverse event data is actually coming from email. It could be forms, for example, like adverse event reporting forms that's been used. It could be also literature. There are a lot of literature cases that really comes in. But if you look at recently, Korea MFDS has adopted E2BR3 as a model to really process safety cases or accept the safety cases, right? But the source data is not E2BR3, right? So how do we really then process these cases? Currently, most of the companies are doing this manually. They would get all these data sources, process them, and then create a safety case entry into the safety database. So this is how they traditionally kind of follow. This takes a lot of time. You need a lot of people to kind of process. Think of a scenario where free text narratives. You have to go through each of the free text narratives to find the specific sentence and what is its relevant information from an adverse events point of view. So this is very error-prone activity. I mean, sometimes human errors really cause those challenges as well. So how do we really then use automation methods to really kind of simplify this process? There are three ways you can really automate. One is rule-based automation. Second one is robotic process automation. And the third one is really using AI technologies. So the first two, the rule-based automation and robotic automation, is more for repeated tasks. The tasks that happens very often, you can really use those techniques. But AI is more sophisticated. It really augments the human expertise in terms of what you can't really see in human eyes. How do we really use algorithms to kind of see that? I'll just give an example of so this is an example of an adverse event reporting form. It's an image that is really received. So how do I then extract specific information? For example, in this case, we are using advanced optical character recognition techniques to extract the ethnicity and race. Only I want specific information to be converted from an image to a text and then processed as part of the safety cases. So all these are now happening automatically. You can really put all your source documents. The system will automatically process the safety cases. So that's really kind of happening as we speak. So the safety signal detection is very, very important to find the risk involved for a specific population. So there are several challenges in terms of safety signal detection. One is, how do we reduce the false positives for signal evaluators in terms of workload, right? That's one. Secondly, how do we reduce the false negatives for patients who are exposed to the particular adverse drug events? And then thirdly, how do we bring in multiple data sets and to really kind of then provide meaningful information based on the data sets? So there are three signal detection methods that are evolving. One, what we call as a multi-model signal detection. Second one is neural signal detection. And third one is predictive signal detection. So these are three major techniques. So in the interest of time, I just talked only the predictive signal detection. So what if you can predict a safety profile of a compound even before you actually start a clinical trial? That is exactly what the predictive signal detection really kind of plays a role. So you can really make very informed research decisions based on the data that you really have and how it will really impact patient lives in terms of saving people when we actually develop a new product. So this is the data that is published in 2020, how it's in Lancet Biomedicine journal that really reported how they can use these technologies from a signal detection standpoint. So the researchers looked at the in vitro data of pharmacology of common targets for more than 2,100 drugs, and they looked at the proteins associated with those treatments. And then they referenced this data with the FADS database. FDA has FDA adverse event reporting database, and then they really kind of see what is the kind of impact, whether they're able to predict the safety profile of those the candidate drugs that's really going to be in the market. So these are nowadays, I mean, these are real life scenarios, and this has been in practice now. It's not just a research paper, but then you really have technologies that's really, in real life scenario. Now come to the last part. So this is again this is the great example of public health surveillance using real-time genomic sequencing. So this has been used across almost 20 countries. This is an initiative by the University of Oxford in partnership with the various other institutes like Public Health England, all of them. So one of the challenges that we faced was how do we quickly find the new variants? I mean, still the pandemic is not over. I mean, we are all taking necessary precautions, but what if there are new variants comes in? How do we quickly find those variants to really look at identify the COVID mutations? That is exactly what this is all about. So GPAS is Global Pathogen Analysis System. It is actually developed by University of Oxford along with other organizations, wherein you can upload the genomic sequence data and you will get the results in 20 minutes. So that's fast it is. It is actually built on a platform called SP3, what they call a Scalable Pathogen Analysis Platform, which is used for tuberculosis. Now they're kind of repurposing that platform to really address COVID-19 vaccine. So the key thing is, this is used by more than 20 countries. So University of Oxford processed almost half of the world's, sequence data. So it really, they really have now volume of data that's really been useful. So this is very, very useful to kind of see which strains are spreading more quickly than others. What genetic features contribute to the increased transmissibility of the vaccine escape. So these are really fundamental things that we really need to look at. This has been gaining a lot of traction after the Delta variant. That's really come into play. So now the last part, I think, I talked a lot about importance of safety and how do we really kind of play. So as we speak, there is significant increase in terms of safety cases, whether it's a vaccine or other products that's in the market. So how do we really augment our platforms, really look at digital transformation of technologies? All of these are really going to be critical for the organizations to really succeed as we move forward beyond pandemic. So with this, I'll conclude my presentation, and thank you for patient listening. Wish you all a very great success for the rest of the day. Thank you.
Thank you very much for your presentation. Very insightful and very informative. Great, thank you. 고맙습니다. 첫 번째 기조연설 함께 하셨는데요. 포스트 코로나의
That was the first keynote presentation. He has talked about the major changes, technology trends in bio industry, especially for post-COVID era. It was a wonderful presentation. Thank you very much. And we would like to move on to the next keynote presentation.